Now that you know all the steps of mitosis, now we're going to talk about cell regulation and how a cell controls mitosis or binary fission, depending on what type of organism it is. One thing you got to keep in mind is that not all cells go through cell division, and I'm mainly going to be talking about mitosis because we're biology and we're going to take a human aspect on this, at the same time or the same rate. Couple of examples. Muscle cells and nerve cells rarely, if ever, go through mitosis. Most muscle cells and nerve cells, you're born with the ones you have, and that's going to be it. In some cases, however, nerve cells or muscle cells can be stimulated to reproduce and to divide. But in most cases, the muscle cells and the nerve cells that you are born with, you have the rest of your life. So if you mess them up, then we have a problem. That's why people can suffer from short-term memory loss or even long-term memory loss, because you've damaged certain nerve cells in your brain, and those will not fix themselves. They will not regrow. Any information that they held is now lost. So that is one of the reasons why head injuries are so bad and why concussions are taken much more seriously than when even I was in school. So never knock your coaches if they keep you out of a game for having a concussion. On the flip side of that, skin cells and stomach cells grow and divide many times over a very fast rate. Our skin cells and our stomach cells are constantly reproducing. They are constantly going through cell division, because just think about it. How much do your skin cells and stomach cells have to put up with on a daily basis? Your skin cells are constantly rubbing up against your clothes. You get cuts, you get bruises. Your skin cells take a lot of force throughout your day. Stomach cells are constantly being surrounded by pretty strong stomach acid. And so they get worn down and they need to be replaced. So some cells rarely, if ever, go through cell division, while other cells, it's just a common day, everyday event that they just have to go through. And it's just an everyday fact of life. So keep that in mind as you are thinking about cell division and getting ready for your unit test. Now, how do cells control cell division? One of the ways that cell regulation occurs is actually a really simple process, and that happens to be touch. One of the biggest regulatory processes is through touch regulation. And this is simply when cells come in contact with each other, they stop growing. And then they'll usually settle down a little bit, and they will eventually go through interphase and then go into mitosis and finish up with cytokinesis. Now let's put this into context. What happens when you cut your finger? Taking what you know of cell division with mitosis and take into account what I just told you about touch regulation, what do you think actually happens when you have a cut on your finger? First off, your cells are stimulated to divide rapidly in order to produce more cells to replace the ones that now have gotten injured, have gotten messed up. Then, as the healing process takes effect and nears completion, cell division slows and eventually stops when cells begin to touch. And that's when you have a healed wound. Another way that cell regulation occurs is through the use of proteins called cyclins. Cyclins are a family of proteins that regulate the progression of the cell cycle. So if you would actually take a look in your packet towards the back, find the page that says cyclins across the top, and it has a little diagram that shows different cyclins and where they would actually come into effect through interphase and mitosis. It is a family of cyclins. There is cyclin A, cyclin B, cyclin D, and E located in the picture. Cyclin D is involved from the get-go. It is there from G1 phase all the way to the end of mitosis. Whereas cyclin E starts ramping up halfway through G1 phase, but then peters out halfway through the S phase. Each one of these has a very specific job within the cell cycle, and it helps control how fast that cycle moves. So they help limit each other. They help keep the cycle steady so that one part of the step doesn't go really, really fast, and then I probably get a lot of boo-boos in that step, and then that just compounds the issue all the way through the cell cycle. So it helps to regulate how fast the cell cycle goes. So to write that down, each step of the cell cycle has its own set of cyclins that increase in concentration to carry on that step, and then decreases when the step nears completion. Along with touch and along with cyclins, you also have internal regulators. These are proteins that respond to the inside environment of the cell. These proteins will only allow the cell cycle to proceed if certain processes get done. So an example of this would be controlling the movement from interphase into mitosis. I have to complete interphase before mitosis is allowed to begin. I have to complete mitosis before I'm allowed to go through cytokinesis. So these are just internal regulators that help respond to what is going on inside the cell and to keep everything in the correct order and not hurrying through one step just to get to another. It slows everything down to make sure everything gets done right. If you have internal regulators, you also have external regulators. And of course, these are proteins that will respond to the outside world of the cell. What these will usually do is direct a cell to either speed up or slow down its cell cycle. So you've got a lot of different things going on. You have touch regulation, where once cells touch, then they stop growing. You have a group of very specific proteins within cell division called cyclins, and there are different cyclins for every single step of mitosis, cytokinesis, and interphase. The whole cell cycle, there are different cyclins that help control what is going on during the cell cycle. Then you also have the internal regulators and the external regulators that will control the other aspects of what is going on, whether it's inside the cell or outside the cell. Now, my last question for you before I let you go is I want you to answer this. What happens when the regulators fail? Think about that one, bring it to class, and I will see you soon.